we will start with the second chapter, that is Shikimate pathway. So far we have seen about the acetate pathway and basics of biogenesis or biosynthesis. Now we will do about the Shikimate pathways. Shikimate pathway, like what we have seen about the acetate pathway. Acetate pathway में हमारा स्टार्ट हमने किया था पहले मैलोनिल कोए के बायोसिंथेसिस से। असीटिल कोए से हमने मैलोनिल कोए बनाया था। वैसे ही यहाँ पर शिकिमेट पाथवे भी स्टार्ट होगा पहले शिकिमिक एसिड को बनाने से। शिकिमिक एसिड जब बनेगा, that will then lead to further other molecules which are coming under the shikimic pathway. So first we will see about the shikimic acid. So shikimic pathway में shikimic acid पहले बनता है, and just like what we had seen it, the precursors for shikimic acid where one it is integrating two cycles, one glycolysis cycle and other pentose phosphate cycle. So it is clubbing together two cycles. One is glycolysis and second as a pentose phosphate cycle. So pentose phosphate cycle और glycolysis cycles दोनों का combination होकर के हमें shikimic acid मिलता है. Glycolysis से हम लेते हैं phosphoenol pyruvate. Phosphoenol pyruvate is the structure for phosphoenol pyruvate will be this will be phosphoenol pyruvate, where it is a 3-carbon compound, 1, 2 and 3 carbon compound. It is having an enol, a carbon-carbon double bond and a hydroxy group, with hydroxyl group is now protected as a phosphate group. So it will be a phosphoenol pyruvate. Plus from pentose phosphate group we have erythrose 4-phosphate. Erythrose structure we know that it is a aldehyde, so it will be aldehyde. Erythrose means it will be having 4 carbon atoms and the hydroxy groups will be on same side, then it will be an erythrose. ये हम स्टीरियोकेमिस्ट्री में भी पढ़ चुके हैं. So erythrose में hydroxy groups दोनों same side में रहते हैं, threose में दोनों opposite sides में होते हैं. यहाँ पे D erythrose होता है क्योंकि शुगर सारे हमारे D होते हैं, L नहीं होते हैं. So D means it will be on the right hand side. So here we will have OH groups on the right hand side. Same है, same side है इसलिए दोनों ही same इधर रहेंगे. यहाँ पर H और यहाँ H and last में आपका it is erythrose 4-phosphate. So 1, 2, 3 and 4th carbon will have a phosphate group. O, PO3, 2 minus. So this will be our erythrose 4-phosphate. Erythrose 4-phosphate and phosphoenol pyruvate. Now in this case what happens, the reaction will be, the first one it will be the phosphate hydrolysis will happen, so that this phosphate group will go out and the electrons comes over here, so that it will be a ketoenol tautomerism will happen, and this then attach the that is carbon of aldehydic carbon. So we will have here it is a 3 carbon and here it is a 4 carbon. So overall we will be having a total of 7 carbon. 7 carbon compound we will get. And that 7 carbon compound we can have this COOH, we can have it as COOH attached to carbon, which is then attached to or is having a carbonyl group as this carbonyl group. Then it will be having a CH2, that CH2, ये CH2 फिर यहाँ पे हाइड्रोक्सी ग्रुप aldehydic कार्बन के साथ अटैच हुआ है, तो aldehydic कार्बन के साथ हमारा रहेगा, that will be having an OH group as it is stereospecific reaction. We will have one OH, then we will have another this CHOH, So here it will be OH. Then again we will have this OH. So here another OH, and then we will have one more CH2, and CH2 then it will be attached with a OPO3 2 minus, that is phosphate group, or we can simply write it as OP. अगर हमें ये इतना नहीं करना है तो this will also be good enough. So OP will be there. So it is now 1, 2, 3, 4, 5, 6 and 7 carbon compound. यहाँ पे 4 carbons erythrose के और 3 carbons from phospho enol pyruvate. We get a erythrose, that is this compound. This compound is known as 3-deoxy D-arabino 3-deoxy D-arabino heptulosonic acid 7-phosphate. 3-deoxy D-arabino heptulosonic we will just eliminate this box. This heptulosonic acid 7-phosphate. At 7th carbon is having a phosphate group, so at 7-phosphate. 3-deoxy D-arabino heptulosonic acid 7-phosphate, or we call this as TAHP. This D coming from here, A from arabino, H from heptulosonic, and P from phosphate. So we'll have a DAHP will be formed. This DAHP will be generated. Then the next reaction will be, ये नाम आप याद रख लो, शायद एग्जाम तक याद हो जाएगा, रट्टा मार लोगे तो बेटर रहेगा. So यहाँ पर अभी क्या होगा? This is OH group. We will also have a hydrogen group. Hydrogen will be here. So this hydrogen now can be lost, and subsequent elimination of phosphate group. So by eliminating phosphoric acid, HPO4 2 minus will be lost. That is, loss of phosphoric acid will give us a enol over here. ये OH group है और यहाँ पे एक double bond generate होगा, so it will be an enol. So that enol will be generated. This enol will be very much similar. यहाँ पे हमारा OH, then we have double bonded oxygen carbonyl, and we have a COH group यहाँ पर है. We have a OH here, and we have another OH here. स्टीरियोकेमिस्ट्री यहाँ पे बहुत इंपॉर्टेंट है. ये स्टीरियोकेमिस्ट्री में चेंज नहीं होना चाहिए. Then it will undergo keto-enol tautomerism. But यहाँ पे keto-enol tautomerism होने पर ये अटैक करता है to this carbonyl carbon, so that we will have 1, 2, 3, 4, 5 and 6 membered ring. And here again the stereochemistry will be retained. जो पहले था enol बनने से पहले same stereochemistry आएगा. hydroxyl group का यहाँ पे OH और यहाँ पे हमारा wedge bonded OH. We will have a here we will have COOH and the dotted OH group. Here we have H and H. This is CH2 group. So H and H. So एक यहाँ से it will be elimination of water molecule will happen, This reaction was an aldol type of reaction. Intramolecular aldol reaction will happen. To give a six-membered cyclic compound. Intramolecular aldol reaction will give us a six-membered cyclic compound. This undergoes dehydration reaction. So that this compound that was formed is called as 3-dehydroquinic acid. This 3-dehydroquinic acid will give us 3-dehydroshikimic acid. Here we will have a double bond OH, then again a dotted OH. This will have an OH और यहाँ पर COH. This compound we call it as 3-dehydroshikimic acid. Oh, sorry. 1 minute. हमारा थोड़ा मिस्टेक हुआ यहाँ पे ये जो जब इलेक्ट्रॉन्स यहाँ रिले होते हैं तो ये हाइड्रोक्सिल ग्रुप नहीं होगा. This will be a keto group. Will not be a hydroxyl, but it will be a keto group. That is why it is called as dehydro. So here it will be a keto group will be generated. This is called as 3-dehydroshikimic acid. 3-dehydroshikimic acid. This undergoes reduction in presence of NADH. It donates H- ions to this reduction. It results in the reduction of this carbonyl, conjugated carbonyl group, and we get hydroxy group over here. This we call it as Shikimic acid. So this is how Shikimic acid can be synthesized by using this particular method, where we are synthesizing Shikimic acid from combination of two intermediates from two different cycles. One is coming from glycolysis, that is phosphoenol pyruvate, and the second one is coming from pentose phosphate cycle, that is erythrose or phosphate. So these two undergoes reaction to form 3-deoxy-D-arabinohexylosonic acid 7-phosphate, that is DAHP, which undergoes loss of phosphate group to give us an enol, and this enol then undergoes intramolecular aldol type of reaction to form a six-membered ring. This six-membered ring then undergoes a dehydration reaction to form 3-dehydroshikimic acid. And this 3-dehydroshikimic acid will then be giving rise to, upon reduction, shikimic acid. So this is how shikimic acid can be synthesized or biosynthesized in the plants. We have to remember here that this shikimic acid or this pathway is present only in plants and microorganisms, not in animals. And that is why whatever the groups or whatever compounds are being synthesized in Shikimate pathway are not present in animals, particularly not in human beings. So we need to have a supply of these chemicals by use from our diets only. That is, from nutrients only we will be able to get the compounds which are synthesized using Shikimate pathway, particularly the aromatic amino acids. So now we will see, as such, Shikimic acid will not be directly utilized for biosynthesis of many different compounds, but it will first be needed to convert into Chorismic acid. Chorismic acid एक important intermediate है Shikimate pathway में. जैसे एसीटेट पाथवे में मैलोनिल कोए जो था वो हमारे काम में आता था काफी सारे मॉलिक्यूल्स बनाने में, प्रोस्टाग्लैंडिन हो या फिर फैटी एसिड्स हो या फिर एरोमेटिक फिनॉल्स हो, फिनॉल्स या फिर ब्यूसियोपल्विन वगैरह जो एंटीबायोटिक्स थे, इन सभी में मैलोनिल कोए की बहुत जरूरत होती थी। जबकि हमारा पाथवे का नाम था एसीटेट पाथवे, लेकिन मैलोनिल कोए हमें वहाँ पे काफी इंपॉर्टेंट लगा। उसी तरह से शिकिमेक पाथवे में भी chorismic acid is very important. This chorismic acid is synthesized from Shikimate pathway only, from Shikimic acid, and Shikimic acid is converted to chorismic acid by first phosphorylation of Shikimic acid. So biosynthesis of chorismic acid. The biosynthesis of chorismic acid, first step involves phosphorylation of shikimic acid. Shikimic acid का structure है, it is a six-membered ring. This undergoes phosphorylation by using ATP molecule, adenosine triphosphate. So this ATP will get converted into ADP, and we will get one phosphate group attached to this particular hydroxyl group. So we will have OP, that is 6 shikimic acid 3 phosphate. We will get shikimic acid 3 phosphate from shikimic acid. Now there will be again addition of one phosphoenol pyruvate. One more phosphoenol pyruvate will get added upon to this. This is phosphoenol pyruvate molecule in presence of EPSP synthase enzyme. This is very important enzyme for plants in Shikimate pathway. If this is not present, then ये आगे प्रोसेस नहीं बढ़ेगा, और ये प्रोसेस अगर नहीं बढ़ेगा तो जो शिकिमेट पाथवे के जो कंपाउंड्स है वो नहीं बन पाएंगे। सो यहाँ पर क्या होता है, वी विल हैव दिस हाइड्रोक्सिल ग्रुप विल बी, यहाँ पे there will be this hydroxyl group lone pair of electrons on oxygen will attack the enol carbon carbon double bond. So we will have H plus ion, phosphate, OH. This will then be attached to the carbon. This will be O, CH2, and the hydrogen that was added. Then we have a OP, and we have a COOH. Now what happens? Here we have a COOH और यहाँ पे double bond. Now this loss of or elimination, 1-2 elimination of phosphoric acid happens. This is also in presence of EPSP synthase. ये एक ही एंजाइम में ये सारा दोनों स्टेप्स एक साथ हो जाते हैं. Again we will have a double bond here and a COH group over here. और यहाँ पे COH. This we call it as EPSP. EPSP means 3-enol pyruvyl shikimic acid 5-phosphate. 3-enol pyruvyl We have 10 more minutes. 3 enol, pyruvyl, shikimic acid, 5 phosphate. That is EPSP. EPSP compound will be formed. This EPSP compound then undergoes elimination of phosphoric acid by 1,4. Here it was 1,2 elimination of phosphoric acid. Now here it will be again elimination of phosphoric acid, but it will be 1,4 elimination. So we have a hydrogen here, a CH2 group. So 2 hydrogens are here. This will undergo elimination. So we will have a 6-membered compound with a double bond here and a double bond here, COOH group and OH here, and we have a O and enol acid. This compound we call it as chorismic acid. It is a simple four-step reaction from shikimic acid to chorismic acid.